Okay, you
Okay.
can do some.
Okay. Thank you, everybody, for joining my thesis defense. I did my thesis at GMIT in Ireland, and there I investigated if there are any complementary effects of using both eDNA metabarcoding and underwater video for marine fish diversity monitoring. So first off, why are we interested in optimizing fish diversity monitoring? Well, because fish diversity and diversity in general has known unprecedented losses in the last decade, mainly to pressures like overfishing, global warming, and pollution. And to combat this, national and international legislation has been made, which requires all nations that are part of it to actually set up management programs to combat this diversity loss. But in order to be able to do successful management, you need reliable data, which you only can get through effective monitoring. And in these last few years, there has been a trend which goes for more non-invasive methods for doing monitoring, as non-invasive methods, as the name suggests, tend to be non-invasive and non-destructive for the environment. And oftentimes they also end up being more labor and cost efficient. One of these recent tools is a molecular tool called eDNA metabarcoding, which uses eDNA or environmental DNA, and that refers to all genetic material presence in bulk environmental samples, and that includes extracellular DNA, which is excreted, but also could include cellular DNA from gametes and such. In general, a metabarcoding workflow starts by taking an environmental sample. On this, you extract the DNA present, after which you amplify a specific marker you're interested in. You sequence the amplified material, and in the end, you perform bioinformatics on your sequence data. At each step of this process, you need to take careful considerations to be able to draw sound conclusions. This is something Emily will touch on in the next presentation as well. Some key advantages for eDNA metabarcoding is that it has a very high taxonomic resolution compared to more traditional methods. It has a very high sensitivity for rare species, and there is actually no taxonomic expertise required for the person performing it. The main drawback, though, is that it is not able to give you population quality information, so it cannot tell you the abundances of the species, or the age, or the sex. So how do we address those limitations? Well, this is where underwater video could be something that could help, because video is able to give you population quality information and can show behavior as well. But its negatives are that it is rare, prone to missing rare species, and is limited by video quality and visibility, and is reliant as well on declining taxonomic expertise. So in theory, both of these methods seem to complement each other very well, yet we were only able to find one other study by Stead et al., which investigated the complementary use of both of these methods. So that's how we got to the aim of our study, which was to investigate the potential use of cabled observatories as multidisciplinary platforms that will use both eDNA metabarcoding and underwater video to get to an optimized marine fish diversity monitoring. We did this by establishing an eDNA analytical pipeline for marine fish in our study area. Second, by implementing an eDNA metabarcoding and underwater video at the same time. And then by comparing the result of both to see if this method is actually feasible. So for the method, first of all, since this project started before I was present on it, everything from sampling up to bioinformatics was done by other researchers. Sampling was performed at the Smart Bay Subsea Observatory in Galway Bay. On the right, you can see a graphic representation of the observatory with, in the orange square, you can see the camera setup. For eDNA, we sampled at 2 time points, one on the 1st of August and the second time point, the 17th. There we deployed 2 samplers, which sampled 50 liters over 24 hours to get both tidal cycles. And for the cabled underwater video, we sampled continuously from July 17th till September 17th, where 2 minutes of every hour were analyzed and all species present were noted down. You can find a live feed of this camera on the website I've shown, and there you can also find all other parameters like salinity as well. Very interesting. Then for DNA isolation, we just used a standard extraction kit. For the amplification, we used the 12S and side B marker regions. We used fish-specific genes to make sure that we find only fish and no other species. But unfortunately, during the PCR step, we had inhibition for all our samples at time point 1, which required us to do a dilution step, but also made sure that we no longer have any undiluted samples for time point 1, which will cause some troubles later on. And then for the high throughput sequencing, we used an Illumina sequencer, which was performed by a company in the UK. Then for the bioinformatics, the first step was doing quality filtering on our raw data. Once we had cleaned up data, we did a process called operational taxonomic unit clustering, or OTU clustering. I don't have the time now to go into detail how it works, but what it does is it turns there are hundreds of thousands of sequences into only a few which are usable. It does it by combining or taking all samples that are 97% similar as one, and then consider this the true sequence. The 97% cutoff is used as it is considered the intra-specific variation of species. Once we had our OTUs, we performed taxonomic assignment to see what species the DNA came from. This was first done in an automated way, as this is a standard way of doing it. And since our data set was 12, we also did a manual taxonomic assignment just to see how accurate the automated version was. Underwater video was able to identify 8 species in total, of which you can see some examples here. For our DNA results, in total, we had 730,000 reads or sequences over 14 samples, which generated 81 OTUs. From identifying these OTUs, we were able to identify 18 fish species. And we can see that 40% of the OTUs were able to be resolved to species level, while 14% to genus and 6 to family. But 40% of our OTUs were not being able to resolve at all. And this is most likely to gaps in reference databases, which is something Christina touched on in her thesis yesterday. So here's a list of our 18 species detected by eDNA with some pictured examples. We can see some economically important species like Atlantic cod. So first, the results I'm going to discuss for the difference in dilution in marker and in time point are generated by the automated pipeline, as this is the standard way of doing it. So we can see that 5 species that we found are unique for non-diluted samples, that 7 overlap in both, and 1 for the diluted. It's not unsurprising that we have more species in the undiluted, as through dilution you can lose rare species. What is more surprising is that there is a species present in the diluted that is not present in the undiluted, as you would expect it to be present in both. This is something that can perhaps happen due to the random sampling, which happens during Illumina sequencing. Another interesting to note that the dilution actually didn't affect the side B marker, but only affected the 12S marker that lost up to 2/3 of its species that it could detect. If we then compare the performance of both markers, we can see that 12S is able to resolve more species than side B. But since there is such a low overlap, a multi-marker approach is actually better than just using one of them. And again, we have to mention that 12S did seem to be more affected by dilution, but it is not certain that this is because of the marker itself or because of other reasons. One reason that side B might have been able to resolve less species is because due to its longer length of the marker, as longer strands of DNA do tend to degrade faster in water and therefore become harder to detect. When we compare to time points, we can see that a lot of more species were detected at the second time point. But again, this is most probably not because there were actually more species present, but it's probably because we don't have an undiluted sample of T1 for 12S. And we probably lost a lot of species due to the dilution step. Then if we compare the automated and manual taxonomic assignment methods, we can see that the automated way find 2 unique species and manual 6 with 12 species overlapping. But the 2 unique species for the automated process were actually misidentifications of non-indigenous species, while it should have been 2 species that are actually present in Galway Bay. And 4 ambiguous OTUs that were only able to be resolved to to genus or family level were able to resolve to species level by doing it manually. So we can see that doing it manually does add a lot of resolution, but it is a very label intensive process, meaning that it's actually only useful for smaller data sets. When we then compare both eDNA and video, we can see that video is able to resolve way less species than eDNA, which is able to detect up to twice as many species. But we can still see that 4 are unique to video, which shows that there are still at least 4 false negatives with eDNA. And we've checked their data in the online database, and they are present there. So gaps in databases is not the cause of these false negatives. I've looked into the behavior, if this could be something, if they were perhaps bottom fish that lived on the seafloor, but this was not the case. I was not able to find any reason as to why they were not able to be detected by eDNA. But the general conclusion from this is that the combination of both methods actually gives a more complete view of the fish diversity present, which is what we were mainly interested in. And so for the main conclusion of this study is that a multidisciplinary approach of eDNA metabarcoding and underwater video results in a higher amount of fish diversity detected. And that with some refinement, this method could be a very good tool for routine fish diversity monitoring. And this could be especially the case at cabled observatories, as it can give you like real-time data. And with now portable sequencers on the horizon, they could be installed and could cause remote monitoring to become a whole lot easier. I would like to thank Luca Merriman for all his help and support during my thesis, and as well GMIT Smart Binary Institute for their infrastructure, as well as the grant that allowed me to do a course on metabarcoding, which helped me out a lot for my thesis. So thank you for your attention. If you have any questions, feel free to ask.